very much all for joining this second sort of technological FRAP session dedicated to spatial transcriptomics. So we'll have this, I think all the speakers agreed, we will have this in English. We have advertised this across all the pancreatic cancer network across Europe. So I don't know how many people outside France we will have, but we've made a lot of, thanks to Norma, a lot of publicity for this webinar, which will be recorded and which will be available afterwards on the FRAP website. So I wanted to thank first all the company that accepted to spend some time presenting their technologies and then discussing the pros and cons that they may have. What we'll do is that we'll have a sort of a block of 3 presentations, and we have 10 minutes discussion, and then the 3 next, and then 10 minutes again. And then we'll have an overview that was made by the Cancéropôle of Lyon, yes, I'm correct, of all the platforms that are available through France. It's probably not, I mean, everyone is buying machine, you know, every day, so it's probably not completely up to date, but I think it's a great effort that they have made. And then same as last time, we're going to have 3 sort of real world user experience that may not be as shiny as, but we'll discuss how, because those data are very nice, but sometimes tricky to either acquire or to analyze. So to keep on time, I think the first, it's the order has nothing to do with anything, but I think the way we've organized this was the presentation from 10x Genomics was first, and I saw that Aurélien is here. So Aurélien, the floor is yours, and you have 10 minutes. And if all speakers can keep their time, then we'll next way we can let you go, hopefully around 4:10.
Yes, thank you, Jérôme. Can you hear me properly? Great. So good afternoon, everyone. I'm Aurélien Bio, Sales Executive at 10x Genomics in France North. And with my colleague, Sarah Milosevic, who is Senior Science and Technology Advisor at 10x, this is our pleasure today to participate to this webinar from the French Research Network against pancreatic adenocarcinoma. So first of all, I would like to thank Professor Jérôme Cros and Norma Blois for the invitation and for giving us the great opportunity to present our spatial portfolio to the FRAP community. Next slide, please. So as you can see, we have a great team in France composed of 9 persons for 3 different sub-territories, France North, France West, and France South. We have different roles, either commercial or technical, or at the frontier of both. And we are working together to help you being successful in your spatial and single-cell projects. So of course, feel free to contact different commercial people or field application scientists, depending on the territory you're located in. Next slide, please. So we'll be focusing today on our 10x spatial portfolio, and due to the time limitation, Sarah is going to give you a quick overview of our 3 platforms, the Visium HD and the Xenium platforms that most of you know already, but also our new spatial platform, Atera, and that is being launched with the first instruments shipped in September. So then I will conclude with a rapid cost comparison of these 3 technologies. Please, Sarah.
So good afternoon, everyone. It's my pleasure to be with you today. So I will move to the technologies and help you to answer the most critical question we can have when we think about spatial is how to select the optimal spatial transcriptomics solution for your research. So to get a good visibility on Visium HD and Xenium technologies, we structured our presentation based on the following framework: assay chemistry, resolution, sensitivity, data analysis, and of course, throughput consideration. Naturally, your specific questions and sample-related approaches are essential to define which technologies or combination of technologies is best suited for your project. So for this, I will also share with you some examples highlighting real cases in the field of cancer research. So first, a quick recap of the two core technology. So Visium HD is a sequencing-based platform that uses slides with two capture areas. It features a continual loan of oligonucleotides that assign transcript to 2 micron squares, which provide single-cell scale resolution. It's highly versatile, supporting FFPE, fresh frozen, fixed frozen, and even archive slides. Visium HD is provided with two formats, 6.5 per 6.5 millimeter and 11 per 11 millimeters. In contrast, Xenium is an imaging-based, non-destructive technology. It uses highly specific padlock probes with rolling circle amplification. This includes a 4-step authentication process to ensure the highest specificity and sensitivity. Xenium provides a large imaging area of 10.5 per 22.5 millimeters and features multimodal cell segmentation to assign transcript to precise X, Y, and Z coordinates. For both technologies, we provide integrated analysis pipelines along with dedicated visualization and secondary analysis tool, Loop Brother for Visium and Xenium Explorer for Xenium. Now, we will have a look at the solutions available for the two technologies. With Visium HD, we provide a whole transcriptome coverage via two distinct chemistries. Visium HD 3', which is a species-agnostic poly-A capture approach, optimized for fresh frozen sample. It can be also used with long-read sequencing to identify, for example, isoforms or to perform spatial detection of B and T cell clonotypes. Visium HD whole transcriptome is here a probe-based approach, which will cover over 18,000 protein coding genes using RTL probes, which are equivalent to our Chromium Flex single-cell assay, optimized for fixed sample. With Xenium, we deliver precision insights by covering up to 5,000 genes. We offer 11 pre-designed customizable panels for human and mouse, but of course, custom panel can be also designed for any species. Researchers can add up to 100 custom genes to any existing panel or design a fully custom panel for up to 480 genes. Now, let's see how the data looks like. On the slide here, we are looking at Visium HD data from a mouse embryo seamlessly overlaid with H&E staining. The distinct color regions represent unsupervised cell clustering, which allow us to identify tissue niches, biological structures. Beyond these global clusters, the platform allows you also to select and visualize individual genes at 2 micron resolution, providing a comprehensive map of the translational landscape. You can as well add on top of this analysis of gene expression for each cluster to perform comparison between cells. We also powered our technology with a new feature in Space Ranger software to support cell segmentation based on nuclei detection and expansion. Now looking at Xenium data. As Xenium is an imaging-based technology, it allows us to observe biological structure at exceptional resolution. First, we use a multimodal approach to perform cell segmentation based on different layers of information: membrane staining, intracellular RNA, nuclei staining. From there, you can then select your region of interest to determine which genes are detected and expressed within your specific area of focus. You can even deep dive to precisely examine individual cells which are detected at per cell level. So now let's see some nice and very interesting examples. The first one I will show you has been done with Visium HD. Here, they used Visium to map the tumor and identify a comprehensive representation of the cell clusters. This provides critical insights into the effect of tumor treatment, specifically to identify hot tumor areas and to characterize the spatial differences between responder and non-responder. The second example is where Xenium has been extensively used to identify cell-cell interaction within tumors and to pinpoint specific immune cell types, such as T cells clonotypes within the tumor microenvironment. Xenium delivers precision insights by enabling you to measure the distance between adaptive immune cells and epithelial cells, for example. The combination of both platforms in the last example can also demonstrate how the combination of the data are very powerful. Here, the authors used Xenium to identify specific cell type called T regulatory memory cells in the mouse intestine with Xenium. And to really gain additional information, they performed Visium assay, which gave them an idea of the cytokine gradients. Taking this data all together, providing them a more complete understanding of cell location and functional states within the tissue. We know single-cell resolution is essential. We know whole transcriptome is essential. And we worked hard to provide you a new instrument giving you the possibility to overpass all these limitations. Now, with Atera, it's designed for unprecedented scales. It enables whole transcriptome profiling at single-cell resolution directly on the slide with a sensitivity equivalent to Chromium single-cell assay gold standard flex. It features a massive sample area over 5 square centimeters, meaning you can fit more sample or map larger sections. It's about more gene, more area, faster results at a lower cost per cells. And before giving the end of the presentation to Aurélien, I would like just to also share with you some example where you can appreciate the capabilities of our platform with the work done at Allen Institute to map human tissues with Atera whole transcriptome. They really demonstrated the resolution and the sensitivity of the platform. Data are already publicly available. So if you are interested, you just need to scan this QR code, but you can also get the information with other samples from our website. And now, Aurélien, you can go ahead with the most interesting part for people.
Thank you, Sarah. So to conclude, here is a cost comparison between Visium HD, Xenium 5K, and Atera whole transcriptome, in which you can see the price per slide, depending on the slide area. And on the right column, you have the price per square centimeter of available sample area. What we can see is that the price per square centimeter of available sample area is decreasing from Visium HD to Xenium, except with the 11 millimeter Visium HD slides where the price is quite similar. Of course, you need to add the sequencing costs on the top for Visium HD, which depends on your sequencing platform of choice. We can see that Atera is the most cost-effective platform with approximately 2,400 euro per square centimeter of available sample area for a full whole transcriptome profiling at single-cell resolution and single-cell sensitivity. So we hope this presentation has provided you with more insights into our spatial solutions. And of course, if you have any questions, we are happy to answer them during the discussion session. And I would like to thank you all for your attention.
Thank you so much, Aurélien. So we'll switch to the presentation from Bio-Techne, and after the last one from Bruker, we'll have this discussion. So I saw that Sergio was here. Is it you that are presenting?
I'm not sure if you are hearing me or not yet.
Yes, we are.
So I'm presenting I have Sergio also with me, but I'm the one presenting today. Are you seeing my screen?
Yes, perfect.
Okay. Yeah, good afternoon everyone. I'm Rasha and I'm field application scientist at Bio-Techne. Today I will give you an overview of the RNAscope technology, its key applications, and also position within the spatial transcriptomics field. And also, I would like to finally highlight the automation possibilities of the RNAscope. Okay. Sorry, it's not moving. So, RNAscope is an advanced in situ hybridization technology. I like to introduce it via this image because it's showing the detection of 12 different RNAs, mRNAs of neuronal markers in one single mouse striatum tissue section. And each dot here that you are seeing is representing a single RNA molecule. So the core of RNAscope technology lies in its unique double ZZ probe design. Sorry. Okay, it's moving alone, I'm sorry. So while the lower region specifically binds to the target RNA, the upper region forms the pre-amplifier binding site, which is required for the signal amplification afterwards. A single ZZ, can bind to RNA, but amplification only occurs when two Z probes bind together, ensuring a very high specificity of the signal. Each probe consists of multiple double Z pairs targeting your RNA of interest. Typically, RNAscope designs include 6 to 20 ZZ pairs per target, so this enables the detection even in partially degraded RNA samples and without any stringent RNase-free conditions required in conventional-ish methods. The second key feature of RNAscope would be the sensitivity. So this is driven by a strong signal amplification system. So once the probe is bound to your target RNA, a cascade of amplifiers form what we call a branching amplification tree. This enables the attachment of thousands of labeled molecules of lower force. And this results in up to 8,000-fold signal amplification, which provides much higher sensitivity compared to conventional-ish. Of course, as each molecular assay, you would need controls. So negative control probes help eliminate all false positive and know the background of the tissue, while positive control probes, those are ubiquitous genes, confirm the RNA quality of your tissue. This is all used for reliable data interpretation of your target of interest. RNAscope is the core technology platform, but we have also several modalities and kits that extend the capability of RNAscope. So the primary RNAscope assay enables you to detect targets above 300 nucleotides, like mRNA, long non-coding RNAs. But in some cases, when you need to detect smaller RNA targets, like 50 to 300 nucleotides, BaseScope is used. This is helpful, for example, in the detection of splice variants. And also, when you do need to detect even smaller RNAs, like microRNAs or siRNAs or ASOs, you would need the miRNAscope kit. So this is, for example, the miRNAscope kit is specifically useful for the studies like biodistribution of your therapeutics. Leveraging the established ZZ probe proximity chemistry, you have seen that ZZ probes are really proximal. So we now offer an assay that is the same concept, like a proximity ligation assay, PLA, enabling the study of the proximity between two different antibodies or proteins. So RNAscope modalities are already widely used across various cell culture systems and also tissue sections. But while some of those protocols have not been validated by R&D, we can still support you with providing data. If I go to the application of RNAscope, so beyond the separate detection of RNA alone and proteins alone, spatial biology now is focused on multi-omics approaches. So this enables the combined analysis of gene, but also protein expression, for example, supporting applications like cell and gene therapies, the study of infectious diseases, or immune repertoire analysis. All of this within the studies that also take into account the functional states using clinically relevant protein biomarkers you already use by or detect by immunofluorescence or immunohistochemistry. So RNAscope can be combined with IF today. This is enabled via a unique pretreatment, which is protease-free, and those conditions preserve both the RNA but also the protein integrity. I would like myself to highlight some of those key applications via this application menu, and I invite you to appreciate the slide and also take a screenshot if you need, because we have now known different categories of applications of RNAscope. You can see 5 of them. First, and historically, RNAscope was used as an alternative for a non-working, for example, antibody or non-existing antibodies. But now it's enlarged to, for example, the study of bacteria or even biomarkers like secreted molecules, cytokines, etc. Moreover, I would like to invite to think about RNAscope when you have therapeutic treatment assessment and biodistribution, for example, studying one siRNA, but also studying the silenced mRNA behind. Most importantly, and this is an emerging use also of RNAscope even more now with the spatial transcriptomics technologies, is the validation of those data. So we have had interest in RNAscope to check really also those data, but also to check visually the quality of RNA before any spatial transcriptomic method, instead or complementary also to DV200. So if I can resume that, we can say that the RNAscope could be already used, for example, during sample selection to assess the integrity of the RNA and confirm the RNA presence. During the spatial transcriptomics run, we have heard about clients using RNAscope following Xenium or Cosmics runs. After the run, to validate spatial transcriptomics data by confirming the signatures of the RNA, and also go beyond and explore other biological hypotheses. This is all done with high throughput thanks to the automation systems that I will present very briefly and the reduced cost per slide in this case. If I need to cite one example for the use of RNAscope in the PDAC context, it would be the albumin detection, for which we see a growing interest. I don't need, I think, to present that to you, but if I quote it, albumin detection by IHC, in this case, because it was challenging as a marker that it's a serum protein and it has caused by IHC high background staining, the RNAscope had overcame this limitation by specifically detecting the mRNA of albumin instead of the protein. We can support you with publications, and here you have a general link for the publications mentioning RNAscope and pancreatic cancer studies. I don't want to go too much onto the details of the HiPlex here, but you also have some HiPlex studies on the COMET platform, and we see a growing interest in the direction of applying also the use of the RNAscope to track also some of the markers all along with proteins studies. So I have also some posters on that. If you are interested, I can share them. I will end with the automation possibilities, just to let you know that the RNAscope historically is a manual assay. So those are the capabilities of the manual assays, but this has been extended over the years using the automation platforms that you know as the Leica Bond, also the Discovery Ultra from Roche, and now also mid-plexing is possible using the COMET that will be presented later by Julien Lavergne. Very quickly on the COMET, it is a fully automated microfluidics-based platform. It does integrate cyclic imaging, but also cyclic detection of RNA and also proteins. It has been built using the patented fast fluidics technology that controls the temperature and the pressure, so it ensures uniform staining of RNA and also of proteins, and it also ensures the tissue preservation and the assay reproducibility. You can also appreciate the fact that the COMET has its integrated microscope into it, and the workflow currently enables the spatial detection for up to 12 RNAs and 24 proteins on a single sample, like in this example, while preserving the tissue architecture. So here you can see that we are detecting 12 RNAs and 12 proteins at the same time. I just want to end saying that RNAscope is supported today by 300 scientists over in the world. From the probe design team to customer support, we have more than 70,000 targets available through already pre-designed, but also custom probes. We can design for any species, any tissues, any RNA. And this method is widely established today with over 14,000 applications, and it is considered as a gold standard for the spatial RNA detection. Thank you all for your attention and also, of course, for the invitation.
Thank you so much. So we'll move on to the last presentation from this block, and then we'll discuss this very different approach, which I think are very complementary based on the scientific question you may have. So, I guess Nicolle is presenting.
Yes.
We can hear you.
Hear me?
Yes, and
Okay.
the screen is full. Perfect. Go ahead.
Okay, perfect. So good afternoon, everyone. I'm Nicolle, the Regional Account Manager for Bruker Spatial Biology, covering France and French-speaking part of Switzerland. So I would like to thank first FRAP Network and especially Dr. Jérôme Cros and Norma for the invitation today. I have 10 minutes to present to you the broadest product line of spatial biology market. So Bruker Spatial Biology is the merging of two companies, NanoString and Canopy, under Bruker entity. So from one side, NanoString brings two special instruments, Genomics and Cosmics, and from the other side, Canopy brings CellScape XR. All instruments have multi-omics capability on transcriptomic and proteomic and developed and optimized for FFPE tissue. Can do more, can do less. So that means, of course, we can work on fresh, frozen, or the tissue type. The difference between each of them will be their level of resolution, we will see that, and their depth of analysis for transcriptomic and proteomic. So let's deep dive with the CellScape XR. So this is the new version of the previous CellScape instrument. There are several ways to make immunofluorescence, but we make the choice to be focused on direct immunofluorescence because it's much more specific, much easy, and flexible to set up high-plex. In this new version, we have 7 optical channels, and it allows the customer to put up to 5 antibodies per cycle. And we are using as well photo bleaching and EPIC IF solution to remove gently the signal from the tissue. So let's focus now on how the CellScape works. It's a cyclic instrument. That means we have 3 steps for each cycle. The first step is staining. As I said, you can put up to 5 antibodies per cycle. The second step is imaging acquisition. The instrument takes the pictures, get the data, and the third step is signal removal, which is, you know, that it's a critical step for cyclic technology. So that's why we are using photo bleaching and EPIC IF solutions to remove more than 99% of the signal in less than 15 seconds at room temperature, keeping the tissue safe and allowing the user to repeat the cycle without limit. So CellScape has the best resolution on the market today with 182 nanometer per pixel. We are using also high dynamic range, allowing to get low and high expression on the same slide. And other important point, and maybe the most important, is the imaging area. Again, we have the biggest imaging area with 44 millimeters by 15 millimeters. In parallel, we develop a complete portfolio of pre-validated panel, labeling kit and service, etc. But maybe the most important, because we are using direct immunofluorescence, CellScape is very flexible, and we know that more than 600 antibodies are compatible with CellScape from AppCap catalog, for example. Okay. So now we can move on Genomics instrument. Again, it's a multi-omic instrument covering up to whole transcriptome with more than 19,000 plex on human and mouse tissue. And we are also able to go up to 1,200 protein on the same slide. It's a biology-driven technology, so that means we are working on biological compartment, and I will explain how does it works in the next slide. We have 5 log of dynamic range and be able to detect low and medium and high expression. We are using a pre-validated panel to same time and keep high throughput. So just quickly, how Genomics works, whether for proteomic or transcriptomic, the system uses the same technology. We will stain the tissue with probes and antibody labeling with oligotag. The first step will be to stain the tissue with one of our panel and up to 4 morphological biomarkers. Thanks to these morphological biomarkers, you will be able to see the structure of the tissue and select your specific region of interest. Then the instrument will illuminate the tissue with a laser, which will release oligotags. The oligotags will be aspirated and put in 96 well plate, and this plate will be analyzed via Encounter or NGS solution. So that means you will get NGS data from each specific region of interest, and for this instrument, the profilable surface is also very important with 35.3 millimeters by 14.1 millimeters. So here I have an example on pancreatic tissue. You all know that on pancreatic tissue, we have islets of Langerhans. So we first stain the tissue with the morphological biomarker to get these pictures and be able to see and select these islets. And we know that in each highlights, we have several cell types, different cell types. And in this case, we used a biomarker to discriminate alpha and beta cells. And each cell type becomes a region of interest and allows the user to get data from each specific cell type and generate, for example, this volcanoplot on the right side. So just for your information, as I said previously, we have several ready-to-use panel. We have whole transcriptome for human and mouse, protein panel up to 1,200 antibodies covering more than 130 PTM. And you can custom each panel with specific antibody, custom antibody or custom probes. So let's move on to the third and the last instrument, which is CosMx. So the CosMx is a spatial molecular imager, deliver unmatched sensitivity, capturing the highest number of transcripts and unit gig per cell. So you see the full biological pictures. It's the first and the only platform to image the entire protein encoded transcriptome inside every cell, a single cell at subcellular resolution. And again, CosMx is a multi-omic instrument and enables stem-cell RNA and protein analysis, giving you a multidimensional view of cell state and fusion. Sorry. What makes this transformative is the ability to image every protein coding gene in a single FFP tissue section. Here we are looking at hippocampus tissue where every cell is profiled at true subcellular resolution across the whole transcriptome. Today, only CosMx delivers the level of spatial and molecular details. The first whole transcriptome dataset was released in 2023, so that means 3 years ago. And even today, CosMx remains the only platform capable to go to whole transcriptome spatial imaging on FFP tissue. Thanks to the direct hybridization chemistry, that scale with plex where amplification technology fails. Another important technology in spatial biology data is accuracy, and cell segmentation is the biggest source of error in spatial transcriptomic. If transcriptomic are not correctly assigned to individual cells, the result will be inaccurate. From the very beginning, when we launched CosMx, we committed to a multimodal approach for cell segmentation. Having a multimodal technology and advancing to 3D is essential. It allows us to define cell by looking at nuclei, membrane, and cytoplasm. Our approach includes advanced training AI with multiple configuration tailored for different tissue and sample types. This combination of multimodal analysis and training ensure we remain best in class for accurately assigning transcript to single cell. Another really powerful aspect is that we are the only imaging, imaging every protein coded gene, but we can also layer protein information on top of that. In this case, we can measure more than 64 proteins. This allows us to look at RNA and protein from the same cells. By doing this, we can explore RNA-protein relationship and orthogonally validate functional RNA target to functional protein markers. This gives us the complete view of both the phenotype and structural information, providing data that neither RNA nor protein alone could deliver. With CosMx, we're giving you a complete ecosystem for single-cell spatial informatics designed to handle whole transcriptome cellular throughput. We build Atomics, a spatial informatics platform, which is a cloud-based analytical solution designed to process, analyze, and visualize whole transcriptome single-cell spatial data. It brings together key features such as high-quality cell segmentation, fast tissue exploration, spatial analysis, and AI tool to help better understand biology. Data format are another key part of our ecosystem. The user can export multiple formats like flat file on the data object. We also provide best practice and pipeline for working with this format, including best practice for optimal analysis. Thank you very much. I hope I keep the time. And if you have any question, feel free to ask now, but you have here my email to ask a question directly.
Thank you so much. Maybe we'll stop here and we'll start taking questions. So if you have a question, please raise your hand. So maybe I'll start with one. So going backward, on the Genomics platforms, because it was really clear for Cosmics. When you have all these antibodies that are tagged, we agree that you get a quantification by ROI. You don't know where they are. Meaning you pick.
Yes, you have first. Yes, you have first to select your ROIs, and then you will get the data from this specific ROIs. So that's, I said, it's a biology-driven instrument. So that means we are working on a specific area of the tissue or specific compartment.
Thank you. Well, then if no one has a question, I keep asking mine.
Or cell type, sorry, just an addition because I forgot it, but as I show in my example, you can as well select antibodies specifically for cell types, and your cell types become ROI, okay? So it's not only a localization, it can be a cell types. Sorry.
Okay, so we have one question from the chat that is asking if we can combine spatial data with ATAC-seq or other epigenomic data.
I can imagine it's a question for 10x because we deliver ATAC-seq single-cell RNA sequencing assay. So at the moment, it's not something we support. I know we have one publication, I think it was like 2 years ago, where people developed in collaboration with 10x specific slides where they performed spatial ATAC-seq, but it's not yet something that we provide. But we are 10x, so we are always working hard to make sure we can get solutions. So I will not have been surprised if it comes soon. But I cannot tell you more. That's just my expectations as well.
Okay. And the following was a second part of the question, which is
due to the dense trauma in PDA.
I'm not sure. So it's for all of us. I'm not sure what's the issue because you have single cell resolution, but maybe Fanny, if you want to rewrite, I'm not sure I understand the question.
Try to identify regulatory programs specially restricted to immune stage in PDAC.
The second part, I think.
Due to the dense trauma in PDAC and low cellularity, what you recommend to control the RNA and Xenium in spatial data sets. Maybe do you want to take the lead, Clara? Clara is our field application, so she will always be good to recommend any potential parts to make sure we get qualification of RNAs.
Yeah, so in general, for all tissues, for Visium and Xenium, we recommend an HNE to control the morphology of the tissue. We also recommend DV200 or RNAscope. You extract the RNA to check the quality to be sure you won't have any bad surprise. And you can even add a DAPI, especially for FFP samples to check the fixation in general. So yeah, this will work for PDAC samples and for other tissues as well.
And me, I had one question for RNAscope, because I think for many proteins, we don't have good antibodies, and this is, as far as I know, the only system that we could almost use in clinical practice. Is how do you, so we see more or less pretty well the boundary of the cells, but how do you recommend that if we wanted to use this in clinical practice to quantify the semi-quantify the expression of RNA in a clinical sample? Did you develop any tools or plugin that could be used in QPAT or in other platforms to quantify the dots that you see from RNAscope?
Yes, I can reply, and Sergio, you can complete if you need it. So for the analysis, yes, indeed, we say that the RNAscope is a semi-quantitative technology. We already have analysis guides with scores of number of dots per cell that could help you with scoring first, and this is the known scores used in publications, so you can confidently use the score. Something else is that we already have some tech notes and webinars and presentations about how clients have used RNAscope to quantify the signal. So clients usually use QPath, they use Omic, they use many other Visium, for example, or Halo. So we have some tech notes, but when needed, if we don't have it, sometimes it's published, or we can give you access to some publications. So it can be quantified when you use those tools, of course.
If I may add something on top of this, because you're right, Jérôme, in saying that historically speaking, RNAscope has been developed for clinical samples. The first use that has been known is the one that led to the development of probes against the HER2 biomarker for breast cancer. And regarding the quantification of the signal, it's been described in the publication from Wang and al. Back in 2012, describing the technology and stating how to quantify the signal. So the principle of signal quantification with RNAscope is based on single RNA molecule quantification. So one dot is perceived as one single RNA molecule. So basically, you need to count down the dots. And either you can do it manually, so old-fashioned, but maybe tedious, right? There are many software-based image analysis out there. Most common one is it's being developed by the Indica Labs company, which software is named called ALO. And there are also, let's say, free of charge software-based image analysis systems such as QPath, which basically works the same, right? You just need to let's say, teach the software how to recognize positive and negative areas, and then the software will subtract by itself the background, and then count down what we call the objects. So you define the size, the color that you need to take into account for the analysis, and then at the very end, you get the macros that you can apply automatically to the same kind of samples. For example, if you're working on the same pancreatic cancer model, you can apply the same macro and then standardize the quantification of the signal.
Okay. And so maybe one last question to the 10X team. The three solutions that you present, how would you sort of qualify the different dynamic range that they offer in terms of a lot, yes, no, or just a little bit, or a lot? Like, how would you rank them in terms of dynamic range for the expression of a single RNA?
Okay, so the power we have at 10X is our Chromium single cell technology. So that's the gold standard where we can really compare with all the other technologies we developed. So if you also look at the way we designed our probes, they are kind of equivalent, like with Flex single cell and Visium. And then with Atera, we also moved on that direction because the building of Atera is with the Xenium basis, but we also took all the advantages from single cell and Visium. And of course, we compared also the sensitivity we've got with Atera, Xenium, with Flex. And this is where we are able to tell you that Atera is as good as single-cell RNA sequencing Flex, where for Xenium, we know V1 is the most sensitive versus V2. So that's how we can allow also a comparison thanks to our single-cell sequencing approach.
Perfect. I'm going quickly. Okay, if there is no more question, maybe we can move on. So thank you very much for all your presentation, and we move to the Takara Bio slides.
Good afternoon, everyone. This is Florian from the Takara Bio team. So I also try to share my presentation. Go to presentation mode.
Yes, we see your screen.
Excellent. Thank you very much for confirming. So also from my side, good afternoon to everybody, and thank you very much to Professor Cros and to Norma for inviting Takara. And I'm really happy to give a little snapshot of the tools that we offer for spatial transcriptomics and actually also for multi-omics. Maybe I have a little answer to the question of spatial ATAC-seq in the end. Takara is quite new for in the context of spatial transcriptomics, but I wanted to give you a little bit of a snapshot of the actually very long history of the company. And from our mother holding, we're a Japanese company that started a little bit more than 100 years ago, actually with alcoholic beverage brewing. So you might, if you like sushi, still order sake labeled with Takara here and there. And branching off from basically having some expertise in fermentation technologies, Takara Bio started very early on in the 60s to supply and design enzymes. And this was basically our path into the biotechnology. And then over the years, a few other companies joined the global Takara holding. The last one being Koryu Biosciences, which joined Takara in 2025, and which is the origin of our spatial transcriptomics tools, where I want to give you a little bit an overview and also tell you a little bit about the history. Overall, we have two solutions available that support spatial transcriptomics and multi-omics assay. The first one is called Seeker, the Seeker Spatial Transcriptomics Kits, which is based on the published SlideSeq technology. This one is a very classical approach. It captures RNA on a surface, then synthesizes spatially tagged cDNA using oligo-DT primers. So it's virtually species agnostic and covering more or less the whole transcriptome. And our setup uses a system that gives around roughly single-cell resolution. So defining unit is 10 micrometer beads in the end. On the second side, and this will be my focus since Seeker is currently not available in France, Germany, and Sweden, is our so-called tracker single-cell mapping kit, which is really a very special approach towards spatial transcriptomics and multi-omics, and based also on a published procedure called slide tags. And here we take a very, very different route to spatial omics compared to Seeker. Namely, by taking tissue sections, we don't disrupt those tissues in the first place, but we actually tag single nuclei in situ in the tissue with spatial barcodes. And then afterwards, we run those nuclei over established single cell or single nuclei platforms that I will mention a bit more in detail later. So we gain true single cell data, and then our spatial image is generated by positioning the nuclei into their original location in the tissue. So when talking about resolution, here our defining unit is actually the nucleus that we sequence. So we don't generate a pixelate picture where you ask resolution in micrometers on micrometers. But we basically really have true single cell or single nuclei data that are generated using this approach. And overall, what is very nice, I think, for starters and maybe labs that want to work quite independently is that the workflow is instrument-free. The technology overall originated at the Broad Institute of MIT and Harvard, and the labs behind are the ones of Evan Markowsko and Chen. And the first technology that they invented was slide-seq, which was then later basically the basis for the Seeker whole transcriptomics kit. And here they used basically an array or a so-called tile of densely packed 10 micron beads that are coupled to oligos that basically are able to capture polyadenylated RNAs. You can place a tissue section on top of such an array, let the RNA diffuse out of the tissue, bind to the surface. Then you generate cDNA with spatial barcodes, and you later amplify and sequence those cDNA molecules. Commercial version is the Seeker assay, where you can see here a little snapshot of how the tiles look in our days. One limitation of such an assay is always that basically you cannot force essentially one cell to sit exactly on one bead, so to give you a really precise single cell resolution. So another question that the two labs asked themselves, is there not any way how we can sequence single cells or maybe nuclei from a given tissue section and give them a spatial coordinate? Basically meaning, can we not take the single data points from our UMAP and put them into a spatial context to generate a spatial image? And the little history is very nicely illustrated also in a podcast that I would like to recommend at this point of time. And it culminated in a second technology developed at Broad Institute called SlideText. And this technology is rather new, so the Nature paper behind it is from 2024, but it also found its way into being supplied to the bigger market, initially by Curio Biosciences and now by Takara. And the assay actually uses the same tiles used for Seeker, but takes the principle, let's say 180 degrees around. So our densely packed beads on a tile, they're still bound to little DNA oligos that have polyase or oligo-DT stretch and the spatial barcode. But in the case of SlideText or Tracker, those oligos are actually cleaved off the beads and migrate into the tissue using a photocleavable linker. So you really twist the original methodology where the RNA diffuses onto such a surface 180 degrees around and release those little spatially barcoded DNA oligos into the tissue where they bind actually to the nuclei. And then we have possibility to dissociate the tissue of the section that was placed on the tile and sequence the transcriptome of the nuclei. And in parallel, by having the polyase stretch, we can capture the spatial oligos. So this is now a little overview on the entire workflow. So basically we start with placing our tissue section on the tile. We illuminate with a short UV pulse. So the spatially barcoded oligos migrate into the section, bind to the nuclei. Then we dissociate the tissue, isolate the nuclei. And then we go into one of the well-established single cell or single nuclei sequencing platforms that might be microvel, like the BD Rhapsody, Droplet, like the 10x Chromium, or combinatorial indexing, like PARSE, for instance, where we generate transcriptome library and then also a feature barcode library that captures the oligos, having the same single cell barcode as the transcriptome. So overall, we have single nuclei transcriptome data, optionally actually also multi-ohm data. So ATAC is something that can be spatialized using Tracker. And on top of spatial coordinates. So our spatial picture in the end is generated by placing the single nuclei from our UMAP into a spatial context of the original tissue. Just quickly mentioning how that exactly works. So overall, a nucleus will in most cases bind a combination of different spatial barcodes from different beads, because lateral diffusion just allows you to basically, or allows the spatial barcodes to migrate quite freely. And then the combination of spatial barcodes per nucleus, this will be something that is unique per nucleus, and it is used to position the nucleus into the original context, since it's known where a spatial barcode was sitting on the tile. So we have two parameters, the barcode sequence and the barcode abundance per nucleus, that the algorithm is taking into account to basically generate the spatial image. Now, to quickly conclude, here's a little overview on workflows regarding the downstream readouts when using the tracker platform. And overall, the system is pretty platform agnostic. So you can see an overview of the different single-cell sequencing workflows that are validated. So this is the 10x Chromium, 3' RNA, but also here the multi-ohm workflow. So there is an option to do spatial RNA plus ATAC sequencing using the setup from 10x, or alternatively the setup from BD Rhapsody. Then Illumina PIP6 is validated. You can generate 5' libraries with a tracker 5C kit. A special one is validated for PARS. And then for people that are interested not only in fresh frozen samples, which applies to those tracker systems, for FFPE samples, we have the tracker FX workflow that works pretty much in the same way, but uses then downstream the flex chemistry from 10x to be compatible with fixed and FFPE samples. So that's it from my side. Thank you very much for the chance and for the attention to everybody.
Thank you so much. We'll do the same. We have these 3 talks blocks. So the next presentation is from Vizgen.
Yes, thank you very much, Jérôme. And I will share my screen now. Yep, share. So can you see it?
It's coming up.
You're seeing it?
Not yet, not yet.
Yes, all good.
Oh, good, perfect. Thank you very much, of course, to Jérôme, Norma, and the organizing people around this platform. Honestly, I come from pancreatic cancer, so I know the need for good tools there. In the end, at Vizgen, we're all about that. That means we want to accelerate spatial discovery at true biological resolution. And we have some very interesting, let's say, platforms to do that. We're a very young company, 2019, founded off the shores of the East Coast in the US. And very importantly, or even more important, we are actually full on continuous innovation with our products, our protocols, and of course, optimization that we want to do. That will be clear in some of the key tones of this presentation. Our scientific partnerships toward the scientific community, being also involved in study design, problem solving, and more, is definitely a statement that we always have been making. And service, white glove service, is something that we take really serious, whether that is with our sales, field team, technical support, R&D, and even our customer support. And I think this is something everybody knows, but it's always good to have a little bit of repetition. We have heard about bulk sequencing, we have heard about single cell sequencing, but these all have, extraction and dissociation of, the tissue context as a consequence. That's also why spatial omics, especially in situ detection, has been named multiple times Nature Methods of the Year. And of course, it's because understanding function requires knowing where cells live and interact. And evidently, losing context means missing mechanisms, missing the true biology. And therefore, cell identity, the neighborhood, and the tissue architecture need to be as much intact as possible, because function emerges from these spatial relationships. And that, at Vizgen, we actually take really to our hearts. That means spatial biology tools that we want to build actually need to cut through that noise, where other technologies might actually provide a little bit more noise due to the typical technical setup. What is really important is to have in situ detection and analysis on a massively multiplex scale, multimodal, subcellular resolution. They need to be flexible and customizable assays. And of course, AI-powered best-in-case image analysis. And that's also why researchers, or let's say expert people, often refer to us as the highest quality data you can generate in spatial. And don't take that from me too much. Take that from the people that have incorporated our platforms, that are continuous users of these platforms for their diverse, robust, and flexible solutions. And it's also remarkable to see also a publication count in high-impact journals. And in the last few years, especially from 2022, we see a stagnant rise of all these typical publications in high-impact journals. But that's not the end. It's, I would say, almost the beginning. We just launched last year the new Ultra platform, so MERScope Ultra. We will talk a bit about that. We launched our V2 chemistry, allowing pretty much limitless detections in also now FFPE. We launched this spring, so really only a month ago, our pre-designed panels with add-on capability designs. And this is really a leverage that is unsurpassed in the spatial field to even fast-track your spatial experiments even more. Also, we launched enhanced cell segmentation portfolios on our VPT, but also, excuse me, in the Vizgen image analysis software. And of course, going into 2027, 20 to 30 marker protein, standard detection, so true multi-omics, and last but not least, 3D. We will expand in a new dimension, thicker tissue on MERScope Ultra, so the current device we have on market. And don't think this is only going to be the beginning. Yes, the beginning, but definitely not the end, because 50 microns is just a proof of study here and is just something what we have done to show that the technology works perfectly. Very simply put, 5 to 10 microns doesn't cut it anymore. That is also logical from our typical biological structures that we need to assay and study from day to day. Therefore, at least if you want to talk 3D, you need to be at 50 to 100 microns. And interestingly, we have shown that in mouse brain, that is perfectly possible with our platform. So true spatial 3D coming, soon to you via the MERScope Ultra platform. So MERScope Ultra, superior flexibility for spatial transcriptomics imaging, but not only transcriptomics, also in terms of proteomics, definitely, as I said, 2 to 30 proteins, due to its 9 auxiliary channels to enable additional stains as well. You can have even combinations with existing SM-FISH technologies, should that be required, making it possible to detect pathogens, synthetic genes, and many more of abundant RNAs or non-coding RNAs, should that be necessary. As I said, the pre-designed panels together with add-on genes or full custom 960 or 1000 plex, including controls, gene panels. And most importantly, the flexibility to integrate with existing pipelines or other data modalities if needed. So what makes it so nice and also so flexible in terms of imaging is that flow cell design. Before that was a 1.25 square centimeter, but still possible if you need only small samples to be imaged. Or you can go to your 3 square centimeter imaging area with the FCXL, typically only available on the Ultra. And of course, you can make it really, completely packed with the tissue of interest or different replicates or naive versus treated tissue. And then it's about choosing your panel. So do you go with a pre-designed panel or do you go with a custom panel? As you see, pre-designed panels can be with you in a matter of just days to a week, which makes an acceleration of your spatial transcriptomics with validated ready-to-run PDPs just an easy to go. Or you can tailor custom panels to your biological questions and discovery goals. And so far we have these panels that have come out. As you can see, pan-neuro and pan-mouse, very typical ones, but also on the transcription factor front of the mouse panels. Also very typically organotypic panels, for instance, colon, lung, or lung wild type as well as cancer panel. And moreover, the metabolic as well as the cardiovascular panels that definitely have already seen some very interesting data. The key benefits of Murphy's 2.0, and definitely one of the most important innovations, is the fact that we can do FFPE without any problems, even with some degradation there. Even with low DV200 scores, Murphy's 2.0 provides really magnificent data on these type of archival samples. So mapping challenging tissues, getting a high dynamic range transcript capture, leading to more cells and more insights, which allows you to identify these types and states in an even more and even less abundant state. That is really what now drives a lot of users towards the MERScope Ultra platform. And of course, if it works in pancreas or PDAC, it should work in everything. That's a bit the statement that I like to use. But truly PDAC or pancreatic ductal adenocarcinoma, especially in one of the typical mouse models here for fixed frozen, is definitely something where they have what they have tested in the San Raffaele Institute, and there they really were astounded by the quality of data. They even call it incredible data also with pancreatic ductal adenocarcinoma. So in the end, as you can see, this is something where we have been working tirelessly on to provide that state-of-the-art, quality data that people want from a platform. But not only them, even across the board, FFP mouse skin, FFP human colon, fixed frozen human brain across the globe, and also Viralis, CRO in France, has shown that from V1 to V2, the results on degraded samples is just marvelous. And that all those data can perfectly go into our analyzer or visualizer, and then into the VPT, where you can analyze all these typical neighborhoods analysis, interaction analysis, and that is completely open to how you actually want to run that. We have also support on that should that be needed. And even more so, we have a team of bioinformaticians via our lab services department that actually can help you as well with those tips and tricks on how to use your data at the best or in the best way. That is also what I wanted to conclude with. Lab services here is almost your direct conduit to some kind of an R&D department or troubleshooting department, where if you have samples and you're not directly so confident, sure to actually start with the MERScope Ultra technology and Murphy's 2, well, just ping us and send your samples over. We can do a consultation, have the design done. We can do the protocol development, so you can at some point really start implementing these kind of workflows at your own lab. You see, due to the size and the expertise in our lab, we can actually manage pretty much every project. Any questions, I would advise to shout that out in the Q&A. Also, if you're interested, I will copy-paste a link to a recent publication with our MERScope technology as well in the chat.
Thank you so much, Matthias. So maybe before finishing, we'll move to the last presentation. Some of those technology, especially for also a single cell analysis, that requires a lot of hands-on times, and there is ways to go around this. They're not free, but and I think the Parhelia presentation will show you some of the ways that can help you fast forward your spatial and single cell data.
Thank you for that. Is everyone able to see my screen so far?
Yeah, it's
perfect. Get loading. All right. So thank you for being here today, and certainly thank you for the invitation. My name is MC Grisillo, and I am the Senior Account Manager for Parhelia Biosciences. I am currently a global senior account manager located within the United States on the East Coast. Parhelia, for a brief backstory, is founded by Nikolai Samusik. He is our co-founder and CEO. He came out of Gary Nolan's lab at Stanford University, and Nikolai's background and really the impetus for starting Parhelia, he was a co-founder of the Codex technology, which has now become Akoya Biosciences and since rebranded or remarketed as the PhenoCycler Fusion technology. But really, his motivation in starting Parhelia, as I mentioned, was he saw spatial as such a budding industry, but that these workflows are very time-intensive, very laborious workflows, very manual. And so Nikolai's vision was to create a completely assay-agnostic solution that is an all-in-one, end-to-end automated sample prep solution, really geared for spatial. And with that began the birth of what we call the Spatial Station. So this is our automated spatial sample prep platform. Its robotic chassis that you see here is based on Hamilton Robotics. So we've partnered directly with Hamilton to build and engineer the Spatial Station. There's two kind of safety guardrail features that I always like to start out with. Number one is that there is capacitive liquid level sensing on board of this instrument, meaning that once that robotic pipette comes into contact with whatever reagent, probe, or antibody that user has on deck of the spatial station, if they do not have enough of that particular reagent to sustain the protocol that they're running, the capacitive liquid level sensing technology will kick in. There will be red lights that flash on the instrument, and a user error message is populated on this UI that you see here on the right side. In addition to the capacitive liquid level sensing, there's a camera underneath the hood of the spatial station. And that camera is there, so as soon as the user selects that they want to run X protocol, they will be provided via this user interface an exact labware deck layout that tells them where to put each module, where to put their pipette tips, etc. But in the event that something isn't seated properly, or if it's in the wrong position, that camera is there to initialize that labware layout prior to being able to press start and walk away, and it will alert the user if something is not in that correct positioning. From there, the other element that I'll touch on briefly is that we have a temperature module on board of the spatial station. I would say that our temperature capabilities are one of our biggest differentiators as opposed to any of the other auto standards on the market, in that we're able to heat as well as cool. So we can go from 0 to 110 degrees Celsius. So for instance, being able to do a 4-degree overnight hybridization, critical in many, many spatial workflows. The spatial station is well suited to perform those 4-degree hybridizations, as well as being able to ramp up to those higher temperatures to automate your DWAX, your antigen retrieval in line with the remainder of your staining protocol that you wish to perform. From there, you'll see the user interface that I've touched on. Really how Parhelia operates is we work very closely with virtually all spatial companies, and we work with these spatial companies to standardize those assays. That being said, a user can turn on the spatial station and they'll see a menu of validated standardized assays that we have ready to go for them. But we can appreciate that each lab likes to go in and do things a bit differently and have some editing capabilities. So with that being said, a user can go into any existing protocol and make edits such as time, temperature, flow rate. If they wanted to add another wash, delete a step, you have infinite programming flexibility to easily go in and edit a protocol as you see fit. In addition to that, there's step-by-step wizards. If someone were to wish to create a custom protocol, by all means, they have the ability to do that as well. Lastly, on the topic of the touchscreen user interface, this system is 21 Part 11 CFR compliant. So with that comes a full run report that is generated at the end of every single protocol that's run on board of the spatial station. So users have a full audit trail of who ran what protocol and all the steps and reagents that were utilized during that run. This is getting into a semi-complete menu of assays that we have standardized on the spatial station. So you can see here we have a dewaxing antigen retrieval protocol. More to say in just a couple of slides on that. This is focusing on the spatial proteomics, given Nicole's background, as I mentioned, being a founder of the Codex technology. But in the transcriptomics world, we have currently many users that are running RNAscope, as well as Genomics. And actually, at the end of last year, we launched our early access protocol to our Visium cytosys. And then we have Xenium in the pipeline currently. Really moving forward for Parhelia, we continue to collaborate, like I mentioned, either directly with these spatial companies, or we find KOLs within these spatial users that are KOLs that are motivated to automate their protocols. So most recently, we've talked with a couple of users that are interested in working together and automating the 10x Atera protocol. So we see that on our horizons very soon as well. At the heart of our technology and where the majority of these staining protocols take place is within what we call our ST12 slide staining module. So this blue box that you're seeing here. And the heart of our technology that we're utilizing is capillary gap staining through laminar flow. And so with that being said, the ST12 module is able to stain up to 12 slides in one run simultaneously. And with that, you are applying our board silica glass cover pad that this blue arrow is pointing down towards. Our board silica glass cover pad has spacers that line the left and right sides. And those spacers are 70 microns in height, which then creates a 100 microliter flow cell. So in essence, many users are actually seeing a reagent savings when it comes to utilizing our capillary gap staining technology within our 100 microliter flow cell, as opposed to manual pap pin staining that can be closer to that 150 or 200 microliter ballpark. This is showcasing a brief blue dye demo to present to you the capillary gap staining taking place, and you can see the uniformity in that staining via the laminar flow technology that I've touched on. Whatever is dispensed across that cover pad is set to hold until the next wash or reagent goes across that slide. Typically, when this is running a legitimate experiment, there is a blue lid that goes across the ST12 module that has 12 corresponding holes. So your samples and reagents are not just exposed to the open light and air. This is just strictly for visual purposes to have that lid off. Another thing I'll mention is you'll see the black pipette tips that were utilized within that blue dye demo video. So these are Hamilton pipette tips that we utilize, black carbon fiber tips, and again, a way that capacitive liquid level sensing technology is present on our system. From there, the other feature that I mentioned I would touch more on is our dewaxing and antigen retrieval process. So I'll preface by saying that the Parhelia Spatial Station is a completely open platform, meaning that users are absolutely free to bring on any of their own reagents. But with that being said, Parhelia does offer what we find to be a superior dewaxing reagent. This is a side-by-side comparison of our Parhelia dewaxing reagent that was done on human brain paraffin-embedded tissue. So this image on the left that you see here was done from a spatial company that was actually shared this image with us using their standard xylene procedure. As you can see, xylene was not all that effective in removing completely the paraffin, and because of that, you can clearly see the autofluorescence as a result. As opposed to when they ran our Parhelia dewaxed solution, they got very clean and complete removal of that paraffin. Outside of our dewax efficacy, the other positive quality about it is that it's completely non-toxic. So there's absolutely no OSHA exposure limits. There's no need for a fume hood. Our dewax is actually from a component that is used in many cosmetic products. So it's safe for human consumption. Outside of offering our Parhelia dewax in a standard bottle format, we also offer it in what we call our Skylab kits. So you see this image here on the left. These are our Skylab kits. There's two versions. Version 1 is our DWAX and antigen retrieval Skylab kit, and version 2 is our DWAX plus H&E version. Regardless of DWAX and antigen retrieval or DWAX and H&E version of the Skylab kit, the idea is that you would receive up to 4 of these perforated 96 well plates that consist of individually aliquoted tubes that have all of the necessary reagents, such as the Parhelia DWAX, your ethanol rehydration, etc. And if you wanted to run 2 slides from this particular kit, you can break away those strips, place them on deck of the spatial station, as you see here on the right side, where we have a plate holder that we provide for you. And then from there, that robotic pipette will go into piercing each of those individually aliquoted tubes and apply those reagents directly to your slide within the ST12 module. This is a very streamlined, standardized way of doing a DWAX antigen retrieval process or an H&E, as opposed to utilizing such bulk volumes of those reagents. Nicolle has said many times the amount of times that he's gone into labs and seen large hematoxylin Coughlin jars with crystals forming in the bottom, and users are wondering why their H&Es aren't turning out so great. This is really solving that issue and, again, bringing more standardization to these processes. This is a look at 6 serial section tissues utilizing our H&E version of the kit. I mentioned that we launched at the end of last year our Visium protocol. So on the front end of that Visium assay, doing the D-cross linking, everything leading up to the H&E, this is routinely done in many labs as they lead into that Visium CytAssist workflow utilizing our Skylab kits. Wrapping up here, the labware auto detection is really what I touched on in the beginning, that safety guardrail feature of the camera that's underneath the hood of the instrument. Secondly, the pipette and gantry auto calibration simply means that there will be notifications to users of daily, weekly, monthly maintenance, where you can have the spatial station calibrate itself simply by pressing a button. There's no need to manually align the X-Y axis. And then lastly, the user profile security, again, coming from that 21/11 CFR compliant aspect, is that users, especially in a core facility setting, could have different accessibility to certain protocols. If a user wanted to be able to edit their protocol and not have anyone else have that capability, you can have those kind of guardrails set up within that user profile security aspect. And lastly, this is just mapping out the dimensions and weight of the spatial station. So as opposed to many other auto scanners that are on the market that are quite bulky and take up much lab space, the spatial station is a true benchtop instrument, about 2 feet by 2 feet by 2 feet. It's quite light, 85 pounds or 35.5 kilograms. We talked through the heating and cooling capability, and then the sample throughput. We have that 12 side staining module, but this 48 number comes from if you wanted to do have multiple ST12 modules on deck of the spatial station for a room temperature protocol. You can have up to 4 of those ST12 modules. Say if you wanted to do a bunch of H&Es, you absolutely have that higher throughput capability. And then the rest with the programming flexibility, again, infinite. Panel mixing is more geared towards our proteomics workflows. Staining volume about 100 microliters. And I would conclude with this is probably the biggest differentiator on the market, or that separates us from other auto stainers on the market, is our dead volume is close to none, as opposed to others where you have to prime a cannula with 200, 300 plus microliters of straight dead volume. You will not see that on the spatial station because we utilize positive displacement pipetting. But with that, I will conclude.
Thank you so much. So we'll open the second rounds of questions. Time decom, maybe I start backwards the same. You mentioned the on the Parhelia platforms you can run the RNAscope, so all the DWACs and then the hybridization and everything. Everything can be run on the station.
Correct, from start to finish. So we have a number of users that are, again, utilizing Parhelia DWACs, antigen retrieval, and then in line with that RNAscope protocol. And we have a number of different RNAscope assays available as well.
So we just put them into and the question that goes with it is that on the 12 slide, can you run different protocols or they all have to be the same?
They do all have to be the same protocol.
Okay. And maybe one, if I don't see any question, one question for Matthias. The panel scalability that you mentioned, because this is one of the issues. Sometimes we only want to detect 50 RNA or sometimes 5,000. But so what's the range that we can sort of design?
So there is full flexibility there. On a standard off the shelf, we have gene imaging kits for 140, 300, 500, and 1,000 genes. But what you can also have, which is a bit of a pity, of course, but you can, for a gene panel, also have 40 spiking genes, for instance. You can perfectly do that with the platform. The unfortunate thing is that you will require to do a 140 gene imaging kit. Now we can be, competitive on pricing there and show some, let's say, support. That's not a problem.
Okay, and maybe I missed you mentioned the Z stack. This is pretty cool, but the X and the X and Y. What's the capture area in 2D?
The Z projection or the XY?
The XY. You said 50 microns for the Z.
So for XY, this will remain the same for the slide. So that's either 1.25 square centimeters or 3 square centimeters.
Okay, 1.25 or 3. Okay.
Yeah. This was just to show on a small size field of view the capacity to do for your single molecular detection in 3D. Because if I show you a 3 square centimeter one, you will not see the granularity of that single molecule, of course.
Okay. And finally, the cell segmentation approach that you use, is that fluorescent marker-based membrane marker, or is it another approach?
There are several approaches. So we have our cell boundary kit, stain kit. This is indeed fluorescent-based, but we also have a poly-A stain that we can do. So there are both ways that can be used. Honestly, that's directly in the hardware of the system. However, our VPT, so our visual processing tool, allows you to do whatever segmentation that feels fit for the tissue at hand. So if there is some curated segmentation approach within your field of research on your tissue, that is perfectly uploadable in that VPT.
Okay.
That allows also you to do very cool bioinformatics actions without the need of a bioinformatician. So you can actually already go to the data without the need to wait, for instance, weeks or months, because the bioinformaticians are not, let's say, there with a lot of people.
Maybe one question for Florian, make sure I got that right. Let's say that we got already some data set generated with whatever your favorite single cell approach, PARS or 10x or things like this, and you want to do some spatial, but you would like to sort of keep the same sequencing technology and its highlights and drawbacks. If I understand well, you provide sort of a nucleus labeling technology, but then you have to go through the classical single cell sequencing, RNA sequencing approach, regardless of the one you want to use.
Absolutely, yeah. So the tracker approach essentially is a kit that labels or that spatially tags nuclei in a tissue section. And then for the downstream readout, this is it's based on nuclei. That's maybe one point to briefly mention once more. But the downstream readout is basically what you know already. So if you're using 10x or PARS or BD Rhapsody or Illumina PipSeq, you would just need, from the tissue section that you placed on a tracker tile, you need to isolate the nuclei, have a good methodology for this. We also have a couple of recommendations and buffers and reagents included in the kit. Might need some optimization, especially for fresh frozen samples, because tissues might be very, very different depending on what you deal with. But then from the isolated nuclei, the downstream workflow is really what you know already. And you can basically choose from all the validated workflows that I have summarized. So you can essentially choose the single cell platform of your personal choice.
Okay, so maybe that's a stupid question, but so to generate the library that will also sequence your tag, you get the same adapters as the different vendors, or you need to make sort of two libraries that you pool?
It's basically using the feature barcoding setup that, for instance, 10x Genomics or BD has available.
Okay.
So that means the tracker barcodes, they are primed also with an oligo-DT primer and converted into a cDNA. Then in the workflow, actually the full-length cDNA from the mRNAs and the cDNA from the tracker, they are actually separated for separate library prep using size selection, so conventional spray beads like MPure. But if you have been doing site-seq at one point in time, or your lab has experience with that, that's essentially exactly the workflow that is also used for the tracker barcodes. I think in the end you can multiplex the libraries again, and then you just need to reserve a certain amount of reads. On 10x we recommend 5,000 for the tracker library. On BD Rhapsody I think 2,000. So you have to just basically adjust the pooling of the transcriptome library with the tracker library to yield a respective amount of reads. And then it's really based on very routine, well-established workflows.
Okay.
I maybe have another question, but it would be for Parhelia. If there is already some experience with gel-based assays, for instance.
I do not believe we have any experience with gel-based assays. The large majority of our users, as you can imagine, are working on paraffin-embedded or fresh frozen, as well as some cell-based users as well. But we have not yet encountered any gel-based.
I was wondering because organoids sometimes are embedded in gels, also in some slides, slide format. So to maybe preserve those domes, that you would cast a gel over that to retain them and then stain. So that was actually the idea.
Okay. Thank you for that. I haven't been asked about that, but I'll certainly talk more with our R&D team about the gel base.
Okay, if there's no more question, and we can move on. Thank you again to all the presenters. And so then you may wonder, where can you do all these technologies on the academic platforms? Or you can, as you have seen, you can send them directly to the company. But if you want to go through an academic platform, I wanted to thank Florence Schaffner from the Cancéropôle Grand Est. I'm really sorry I said the Sud-Est, but it's Grand Est, who led an effort on spatial biology, and we'll show you, the effort of mapping all the platform. You can do all those approach. Thank you, Florence.
Yeah, thank you. Thank you so much, Norma and Jérôme, for inviting me to present this emerging network. So it will be another network supported by INCA in a couple of months. So it will be especially focused on spatial omic. And I present that, but I would like to say it's an effort carried by the 7 Cancéropôle. So if you say Cancéropôle Lyon, it's also okay. It's not where I'm from, but everybody from each Cancéropôle is working on this emerging networks. So we as Cancéropôle, we had all actions on spatial omics, mainly transcriptomic, and we decided to work together to support this development in France. And we identify 7 experts, one in each Cancéropôle, and we get together, and it was rapidly, the conclusion were rapid that we need to structure at the French level this rapid and fast development. So we identify 5 working groups, so sample preparation, transcriptomic by imaging and sequencing. So we spoke already of these 3 working groups today, spatial proteomic and data. So what we did is that we identified experts in this field already working on these different techniques, and we put them together and make like a big brainstorming on what we have to do, what is important, what is needed to move forward. And one of the things was we need a map. So we need to know what is available where, because France is large, so the Cancéropôle covers the whole territory. So and we know pretty well the ecosystem, the research ecosystem in France. So we did a census in all our research teams. So who is doing what, and defining by our working groups. And this is an update of March 26th of this year. Obviously, it changed rapidly. Not so long ago, there was no Atera. Now there is an Atera, just to mention the very first machine that was explained by Tenix, but there is many other rapidly moving companies that are coming into the market. So at the moment, that is what we have. And I focus on because today it's a transcriptomic webinar, I focus on these specific techniques. So we identify 25 platforms all over France. So you see it's quite well dispersed in all France. And you have the list here. So maybe you are not on the list. So if it's the case, please contact me or one of your Cancéropôle colleagues. I have put the name at the end of the presentation. So this is what we have for sequencing and imaging. It's less platform. It's only 16, but some are doing both, of course, imaging and transcriptomics and sequencing. So we also did for not all the equipment, so I can see already that we have also to update the equipment list that we have been asking. But that will be for the future network that will be put in place in 2027. So the objective of this network of spatial omic is really to integrate all the omic possibility. So from transcriptomic, of course, but proteomic, metabolomic, epigenomic, what do I know, all these new omics that are coming forward. And the idea is to identify them, that people can find what is done where, and how to contact the people that are doing the work. But also some guidelines and good laboratory practices, coordinate and promote open database and tools. Many labs are developing tools to analyze this data, and it's very important that this is known to everyone, that they can use them. Obviously, we also do a lot of sessions for formation. There is some expertise that don't have at all any summer school or webinars to inform people how to use this technology and what they can bring to their work and their science project, scientific events, and promote innovation and technology development. The idea is really to accelerate the integration of all these spatial omic into translational and clinical research in France, like I think we're going to see some examples coming up in the next presentation. So this is what the network will do. This will start in 2027, as I said, supported by the INCA, the French National Cancer Institute. And here is the name of all the experts, because I'm working at the Cancéropôle. I'm not an expert in the field, and we have the expert in each Cancéropôle that works with us to build this network, and that to make sure it's going to bring what the researcher and scientists need to operate this new technology and use them in the better ways to get the most meaningful from the data. So keep in touch. You can contact me. I put my email, but contact any of us. This is all the same. And we are looking forward to you to participate and come to our network in the FRAP network, but all this other network from INCA and others institute. So that would be it for me. Thank you.
Thank you so much. I think this is a very, very important initiative that we know where, and we coordinate the efforts, especially for analyzing those data, because it's, you know, one thing to generate them. It only takes money, a lot of it, but only takes money.
Yes.
But if you underuse them, it's a pity. And this is the very difficult part, is to actually analyze those massive data and try to get the best out of them. And so if we can share those experiences, I think this is very important.
Yes, this is the idea. There is at least 3 working groups just for data, because as you said, it's crucial, and it requires a lot of brain and computer power. So it's very important that people get together and share what they're doing, that things are moving faster like that, for segmentation, for analysis, and so on and so forth. So keep in touch. There will be a call for proposal in the coming weeks, and then starting 2027, will be up and running, but we hope to start slowly but incrementing with all of these things going through the year. Thank you very much for the opportunity, at least.
Thank you. So to conclude, we'll have, and I wanted to thank all of them, 3 power users of different technology that will show you on some data sets the ups and downs of them. And we start with Rémy Nicolle in Centre de Recherche de l'Inflammation à Paris to discuss some of the data he's been using on Visium. So the first generation, but that I think a lot of us have used, and what are the ups and downs. Thank you, Rémy.
So, hi everyone. I'm supposed to show you how we struggle with those data. All right, so we started a few years ago, and we're actually going to talk about the Visium and the whole transcriptome techniques. Just a quick reminder, at the time, what we had to do is we had to cut little pieces in the slide and put them on this 4 different pieces of tissue on this one slide. And we had spots of about 55 microns on which we could sequence the entire transcriptome. So this was the first version. And what we did is that we did this on archival paraffin-embedded tissues. So like whatever Jérôme has sitting under his desk for the last few years. So old tissue, that's an important part. And then later on, we tried the cytosist, which was supposed to be easier and faster, etc. Actually, so the first thing I'm going to talk about struggling. I'm going to talk about all the problems, not the good parts. That was the previous discussions. And so we actually had this first, so only FFPE, and we tried the first kit, which was called V1, and the second, which was based on cytosist. And what you can see here are the numbers of samples we did on the first version without cytosist. So 12 samples and 16 samples on the cytosist platform. And the failure rate is quite high for Visium, again, on old paraffin-embedded archival pathology tissues. These are not the numbers you would get on mice, for instance, or on very recent or even, of course, frozen tissues. Another struggling part is that it's not trivial if you go through a typical sequencing platform to actually process these, because there's a lot of requirements on how the files and the naming requirements are quite stringent. So I advise you to go through a very typical platform that do a lot of Visium, basically. Okay, so what kind of problems do we get? So this is the kind of tissue image you get. And the first thing that actually happens quite a lot is that you're missing an entire section of tissue for which you have absolutely no data on the Visium. This is quite frequent. Another part is that you're looking for some very specific tissue, and it's quite the opposite. Well, this is just a worsening case of you only get the transcriptome on some very small part of the tissue, and sometimes the tissue is so degraded that actually the assay does not work at all. And this is typically what you get when you have very, very few genes, so few that the spots are not really, you cannot characterize them with whole transcriptome. Now, those are all the bad parts. I have to say that when it works, it's quite beautiful. So these are the different types of stroma that we have on PDAC, and it really quantifies very well the different regions of the different types of stroma, where they are, how they interact with different types of tumor cells. So it is very nice. Now, if you go to more precise, higher resolution techniques, so we tried mostly Xenium, and I know there's going to be a better presentation in this, but I would like to highlight some of the problems that also apply to actually Visium HD, which is the only thing available now in the Visium technology, as I've understood. The first thing that we always have with those high resolution methods is really a very, very difficult cell boundary identification. So what we call cell segmentation, it is not a solved problem at all. And this is true both for image-based or sequencing-based. So I'm not sure it's going to be solved anywhere soon, but I do have to say that most of the differential expressed genes you can find are actually the neighbor's gene. So it's not trivial. So this is typically what you try to get as a better segmentation. Keep in mind that the segmentation has to be 3D, which is extremely difficult, probably not feasible in some cases. And so what you can do is you can test the purity of every single cell, spatial single cell, using a reference real single cell atlas. And from those, what you usually get here is quite a lot of, here it shows on the bottom, quite a lot of cells that are not pure. And not pure can mean more than 20% of the neighboring cells. So the transcriptome is not perfectly single cell. One last thing, and I'll conclude with this, is that we all want to do niche analysis. So what are the typical types of cells that do interact together often in some patients, not others, etc. So here's what you want to get with high resolution transcriptomics, a niche here that can be mixtures of different types of cells that would be enriched in specific cells. Now, if this is what you really want, actually Visium non-HD is perfect. It really gives very robust niches. So what I'm trying to say here is that keep in mind, what do you really want? If you want true single cell transcriptome, spatial is not trivial for this, or spatial imaging or Visium HD, etc. If you want niches, actually Visium non-HD or something more coarse can be sufficient. I'm just trying to say that keep in mind what your goal is and adapt the technology to it. That's it for me.
Thank you so much. And just to clarify, the tissue we provided to Rémy, they all passed the DV200 QC, so we didn't give you crap. But it's true that we did struggle a bit more with the second generation that we had with the first one. Do we have question for Rémy on how do you start to analyze those data? Or
maybe
no,
I just would like to mention that we do have a platform that is called the cloud, where you don't require any bioinformatic experience to run a Space Ranger. So you don't need to run this script on a Linux environment, and it's free and you can use it as you wish.
Thanks, that's good to know.
You're welcome.
Okay, so if there's no more question, we're moving to Julien, which is at the CHIC platform in Paris and has some extensive experience on many Omic technology, but he will speak to us today about the using of RNAscope and multiplex RNAscope.
Yes, so just I will share my screen. So yes. So hello everybody. To begin, I'd like to thank the FRAP and the organizing team for this invitation to talk with you about RNAscope, and give you a feedback from our platform SHIX. So SHIX stands for Cordelier Histology, Imaging, Cytometry, and Spatial Omics. And we are localized in the Cordelier Research Center in Paris. And I am Julien Lavergne, responsible of the transversal projects within the histology and spatial omics facilities. So the goal of our platform is really to help our users through their scientific project by giving them our expertise in sample preparation, developing their mono to multiparametric staining, their quantitative image acquisition, and also their data analysis. So if you want more information, you're free to go to our website to see all the instruments that we possess and also our emails contact. So the rest of the presentation will focus on the application of RNAscope in the SHIX platform. And to do so, we have 3 instruments. The first one is the iBase2 instrument to perform the hybridization of RNAscope of a manual experiment. And here I just repeat the workflow that you might have heard of and you've heard in previous presentation. And in the SHIX platform, we currently performing mostly the RNAscope HD Brown kit, which is the one with the chromogenic revelation, but you can also perform fluorescence protocol. So concerning the feedback, well, the only good feedback I could provide is to really pay attention on the pre-treatment of your sample because it's depending on your tissue. Apart from that, well, the protocol is quite easy to follow and there is no major issue about that. So that was for the manual staining. And now for the automated ones, we have 2 instruments. The first one is the Leica Bonderix, which allowed the protocol of mono to middle multiplex of RNA detection and on whole slide staining. And we mostly use the Multiomics LS assay kit, which is also allows the co-detection of protein and RNA on the same slide. So the protocol is the same with the famous ZZ technology. And what will differ is on the revelation. Here we're using the pyramid system amplification, and it relies on adding a secondary antibody, HRP, against a channel or markers, and it will react with the pyramid by creating covalent bonds near the markers with the fluorescence signal. And this is where really you will have the optimization. So what about the recommendation? What you have to know when you perform RNA experiments is to have for each of your sample 3 independent slides. One will be for your control probes positive in order to qualify the RNA of your sample and also to qualify and to validate the workflow of your kit inside your instrument. Another slide is for the control probes negative in order for your analysis by removing the aspecific dots you'll get on your sample and then your slide with your panel. So with the kit we are using, you can go up to a 6-plex maximum. And in the SHIX as fluorophore, we are using the Opal from Akoya, now called Quanterix. And where you have to pay attention is really on the pairing between your markers and the fluorophore. By that, I mean to attribute to the high express marker the fluorophore with the lowest signal intensity. As for the acquisition, you have to be assured to have access to an imager compatible with the Opal. And here at the SHIX, we have the PhenoImager HT 2.0 from Quanterix, specifically designed for those kind of fluorophore. So when you're performing a RNA, you have to use preferably the objective at 40x. And because this technology relies on the spectral imaging, it is better to have a spectral library for your own sample and also an autofluorescence slide to remove the autofluorescence from your tissue. So both of those slides are not mandatory, but they will improve the acquisition and the data analysis. So as an example, on those kits which were running on the Bonderix in our platform, it came from a user from a Cordelier Research Center, and we work on FFP section of human colorectal cancer. And the experimental design was the following one, with 4 RNA and 2 proteins that were already optimized. And that is a strong recommendation. Before starting a multi-omic protocol, you have first to optimize your protein. So the workflow was with the 3 independent slides that I mentioned before. And for the protocol, well, we arrange a classical sequential immunofluorescence for the protein post-RNA detection. And this is actually how it works mostly in the multi-omic experiment. First the RNA and then the protein protocol. As key data, well, the run was 24 hours for 20 slides, and the cost was approximately 600 euros per slide. And this is the image that we got. So it was at 40x and on the control probes positive. So here we can clearly see the dots in orange, in white, in red, and also the protein markers in cyan blue and green. Sorry. So yes, and now let's move on to another instrument, which is the Comet from Lunaphore, which allow us to perform a higher plex on RNA detection. But compared to the Bonnerix, here we work on region of interest. And the region is 12.5 millimeters per 12.5 millimeters. And we are using the RNAscope iPlex Pro kit. So this instrument relies on a cyclic experiment, and it's not only an autostainer, but also an imager. And the heart of the protocol is right here. You still have the double Z and the amplification tree. And here you have first the fluorophore hybridization, RNA imaging, and cleaving. And then you restart this or redo this cycle n times until you go up to your plex. And then you can also add a protein detection after your RNA. So as for the practical recommendation, well, we still have the same as for the manual or even the other autostainer, 3 independent slides. And here, as I said, you can go with a higher plex, which is 12, twice the amount that compared to the Bonnerix. And also you still have to pay attention to the pairing between your express marker and also your fluorophore. But here it's kind of different because on the Bonnerix we're using pyramid system amplification, and here it's more like classical Alexa fluor secondary antibody. Concerning the template, well, it's quite easy to use on the Comet. Everything is well written from the pre-treatment to the image acquisition, and you just have to click, so really well written and easy to perform. So let's finish with an example on an experiment we perform on this instrument, which is also a multi-omic protocol from a user from the Cordelier Research Center on FFP section of biopsy from patients with rhabdomyolysis. So the experimental design was the following one with 2 RNA and 20 proteins. Same workflow with the 3 independent slides. And for the protocol, so as I mentioned, the system is also an imager. So, and here you have to first write your exposure time before starting your protocol. And it's kind of weird and it's kind of tricky, but here for this experiment, we use the Lunaphore recommendation, and it worked really well on the first experiment, but I cannot assure you that it will work as good as this experiment if you change the target or a sample tissue. And as key data, well, the run was 30 hours for 2 slides, but I would like to remember that this is on biopsies and on region of interest, not on a whole slide staining. And the cost was 1,000 euros per slide. And here you have a pretty good image of what we got. So here you have an exhaustive image of the protein markers. And here at the bottom left, the RNA dot detection. And it was really good. The user was really happy with what he got. So I'm at the end of my presentation. So if you want more information, more feedback, or if you want to perform your experiment in our platform, you're free to contact us. We will be very happy to help you. Thanks for your attention.
Thank you very much, Julien. And does anyone has question? For all of, I'm waiting they come, for all of those technologies, sometime on the fluorescence or only on cell segmentation, it's pretty hard to know, not always easy to know what cell type you are dealing with. with the RNAscope, I guess in what shape is the tissue after? Can you do an H&E, or should you do an H&E before if you want to stack them to know exactly who is who? Is it possible?
Yes, you can perform an H&E after this kind of protocol. Yes, it damage a bit your tissue, but it is preferable to have an H&E on a sequential slide.
Serial section.
Yes, but you can perform an H&E. Yes.
Okay. Julien, could you permit me to add something to that?
Yes.
So about that, we have indeed for H&E experiments like on the Leica bond and the Discovery Ultra clients doing H&E afterwards. The limit may be on the Discovery or the Leica, the fact that we are stripping also between antibodies, and that helps sometimes in the degradation of the morphology, but still some of my clients are doing H&E afterwards. And regarding the comet, on the comet, it's a little bit different because of the fluidics ship that we have mentioned and Julien have mentioned. The tissue morphology is more preserved, okay, because there is control of temperature and also of the fluids and the pressure. So usually clients are able, even after 80plex, doing H&E after a comet run. So that's less limited on the comet in this way. Thank you.
Okay. Guess everyone is sort of killed by the heat. So maybe then we move to the last presentation. Thank you so much to Zeynep from Corinne Bousquet's lab to talk about her experience on Xenium, which was, I believe, also held by FRAP. So we're really happy that we were helpful in helping you to go, I think, abroad to study those data. Thank you.
Yep, thank you. I will share my screen quickly. Seconds. You see it well?
Yeah, it's not in full. Yeah, perfect.
No? Okay.
Yeah.
Let me get my... Yeah, so first of all, thank you so much for the opportunity to speak. I'm Zeynep, a PhD student, and I've been working with Xenium for a while. So I'm going to tell you about my experience with it. First of all, I'm going to tell you about what I could get, the results I could get with the Xenium, and then we can move to the QC and disadvantages. So first of all, the description of our run was that it was run on a Xenium slide of FFPE with 8 TMA cores, as you can see here. So you can indeed perform here an H&E before to see all the morphology you want. And you can even annotate it and align it with your Xenium image after. So you can see the transcripts on the slide, H&E. So as our study was more targeted, we selected 400plex pan-cancer gene panel. This was pre-designed. And we added on a 100plex gene panel that we customized. As it's imaging-based, I will add. And also we used multiplex segmentation staining kit, as we know that segmentation is always an issue, and I will talk about this later on. So first thing I did, as it is a single cell resolution technique, I could treat it as a single cell sequencing data, essentially. I have very little genes for that, but yes, still. I can do a predynamic clustering. So what I did was to cluster my cells, and then I ran an essentially differential gene expression on these cells to find the cell types. So to my best ability, I identified some cell types and annotated them. This is challenging when you have a little, not so big of a gene panel, I will say, but now I know that 10x has a 5K panel available if you're working on more cell identification studies. So The first thing you can do is a distance analysis, as it's a single cell, as I mentioned. So what I did here is to extract my cell types of interest, so 5 types of fibroblasts, let's say, and tumor cells that are classified into basal-like and classical. So you can do calculation of the closest neighbor, the distance to the closest neighbor, and compare them. Very standard. What you can do also, as Rémy mentioned, is a niche analysis, kind of. And this is more accurate, I suppose, from Visium because you have single cell resolution. So what I did here is to take a cell in the middle, so my CAFs of interest, fibroblasts of interest, and then I counted the other cell types, as you can see here, adipocyte and endothelial, as I annotated before. And here you can see an image from the Xenium Explorer software. This is available in the 10x platform. And it gives you quite high quality images that you can visualize your transcripts or your cell types, as you can see here. And then there are many packages to do this. You can see how in the niche your cells communicate with each other. So this is a ligand receptor pair analysis. And you can see here from the CAFs of interest, fibroblasts of interest here, the ligands that are secreted and then the receptors in the neighbors that receive these ligands. These are some well-known packages to do this. So if I move to the quality control, 10x gives you a big list, a big summary of the quality control. So if we start from the number of cells detected, this is from one core of the TMA, by the way. We had 110K cells, of which I identified almost 30K fibroblasts, so which is reflective of the real thing, I think. We had 130 median transcripts per cell. I will talk about it also later, and a high number of high-quality transcripts, so that's good. To go into detail, we had a lot of cells that had transcripts, but almost 20% of the transcripts were outside of a cell. And this could be because of the segmentation issue. It's a considerable number of transcripts that are outside of a cell, but they are not taken into account anyways because they are not segmented. So as I said, the median transcript number per cell is 130. And I saw that in average, you will have 200 to 300 in an in situ hybridization method that is image-based. So it's a bit low, but it might be also because we don't have a huge panel. And yeah, so it's acceptable, let's say. And percent of empty cells is very low in all my samples. I don't have this problem of empty cells. And even if I do, these are filtered out anyway in the processing. So to check the transcript quality, you can see it here in the plot that all the pre-designed and the custom genes that we picked are high quality transcripts in the sample. The only low quality ones are the negative controls, which you would expect because you don't have them much in the sample. And here you can see where you have these negative controls located in the sample. You can see that it's in the edges of the tissue and not so high in amounts. And we also had a very low false positive possibility estimated number. So that was also good. So I think the biggest problem we have in spatial methods is the segmentation, as Rémy mentioned. So for this, 10x offers you an optional improvement, let's say, the multimodal segmentation kit. These are the stains that it uses. So we did indeed use this to improve the segmentation, sorry. So I'll give you an example. On the left, you can see the actual staining on the tissue of these multimodal cell segmentation staining kit. So for example, if it's pink, it's a boundary stain. If it's yellow, it's an interior. And here you can see the cells segmented on the right. So if it's segmented by the boundary stain, it will be pink and so on.
And we can see that here, for example, you have an overestimation of the cell number. Here you can see one cell clearly from the nucleus, and it's detected as 5, 6 cells. So this is quite commonly seen. Another thing you can have is that you can miss areas of cells. So here you see that there are some fibroblasts. I will say that especially if you're working with cells that have elongated shapes or adipocytes that are quite empty, normally you will have this problem. If you're working more with epithelial cells, you will have a less of a challenge, let's say. But you can have areas of cells that are missed because of segmentation issues. So this is the conclusion of my presentation. I think the biggest issues are the complexity of your panel, but this is up to you if you're more discovery-based studies or more targeted studies, you can choose this. And the segmentation. Thank you for listening.
Thank you very much. While waiting for question, one that I have is if you look at, for instance, genes of epithelial genes or fibroblast gene between the positive and the mildly positive and the high expressing cell, what's the difference in terms of number of transcript per cells? Are you looking at 0 to 3, 0 to 10? What's the range?
Yes, so for example, collagen genes for the fibroblast, you have a high number of transcript per cell. So it can go up to 30, 40, I think, even more, I think. So you have a good, you can clearly differentiate between epithelial and mesenchymal, for example. But if you're really looking into like subclustering, subtyping, it's more like you have to have more bigger plexity.
And one question in the chat is that how much time did it take you to get all those results from the acquisition to get used to the software, cell detection, and so on?
I will say that I was not involved in the sample prep, so the HNE or the making the TMA, I was not. But the platform was quite fast, I will say. I think we got the results maybe 1.5 months after we sent them the sample. If I'm wrong, someone will fix it.
I'm just thinking, Clara, would you like to give an idea of the workflow, how many times it takes from the sample preparation to getting the data?
Yeah, so it's a 3-day workflow, and then you put it in the instrument. It depends on the size of your samples, but for a 5K in general, it's around 5 days, and then you don't have to sequence anything. It is ready to do the secondary analysis. The primary analysis, the clustering, etc., is already done in the instrument. So yeah, in 1 week, you can have almost everything.
And I just have a question for you, Zeynep, because you mentioned you had issue with cell segmentation. I was just wondering if you tried with third-party tools such as CellPose. I know there is some people trying to get it like this. What is also interesting for me is to know how many cells were segmented based on the different multimodal approach, because you mentioned and you shared an image, which is also a quality control we take depending on how many cells has been segmented with all these steps, because it's an algorithm taking step by step the segmentation. And usually when we have a lot of cells which were segmented based on the nuclei expansion, it means also that at one point we can think about a tissue issue. Or at least if Clara, you want to add on top of that some part that you also observed with the Xenium runs and the cell segmentation.
Sorry.
Yeah. Go for it. No, you first and I complete.
For your first question, I know that we can use multiple tools for the segmentation. I didn't do it because I thought it compensated between missing cells and overestimations. So I did not do it, but I know you can. For your second question, I think the nuclear expansion with DAPI was a very low number of very low percentage in my samples. So it was more in interior staining or boundary staining.
Okay, because we also have users with specific tissue types where the multimodal approach can be tricky, even if it's a good solution. So they can add on top of that IF staining, which can be used after the Xenium run, which is a non-destructive one. So you can always think about running Comet, for example, or Akoya system. And then you can use these markers to also work on cell segmentation with third-party tools. So we know there is samples which are tricky. Unfortunately, this is not always depending on the sample prep. It's also the sample complexity. And there is always a way to bypass. So if you need any additional info, do not hesitate to ask the 10X team. We always have good tools to share.
Okay, so thank you so much to everyone. We are not as late as last time, so it's almost fine. So the whole seminar will be available on replay on the FRAP website. So you will have a few days. So you're welcome to come and relook at your specific part, and you will have all the contacts of all the companies. So thank you again to all the speakers that spent the afternoon with us, and wish you good luck to all your endeavors in spatial biology. And we will probably organize the next technical webinar with maybe probably be on single cell. We'll see, depending on the interest of the community. So once again, thank you so much to everyone, and have a good end of the afternoon. Bye-bye.
Bye, thank you.
Thank you.
Thank you very much. Bye.
Bye, thank you.
Thanks.